Okay, everybody. So I want to go through and hit the highlights of, or I should say, highlight the main topics of Chapter 8. Looks like YouTube cuts me off about 8 or 9 minutes, and that's when I usually lose most of, your attentions anyway. So I'll just go through pretty quickly. Just like Chapter 7, I just want to point out the big ideas here. So cell rest, you could think of it as being the opposite of photosynthesis. It's not necessarily the exact opposite. But in photosynthesis, we were taking energy and storing that in a solid substance. Cell respiration is the way that a cell, through the mitochondria, can take that solid source and get at those electrons, pull them apart, access that energy step by step by step. That's why it's an oxidation reduction reaction. In one portion, we store the electrons, the other portion, we pull the electrons away, because you can't just let those electrons go. You'd lose them. We learned in Chapter 2 they're too small. We can't see them. We don't know where they are really. We have an idea, right? Mathematically, we know where they exist in those energy levels, but we can't view an electron. We'd lose it. So anytime you remove that electron energy, what the cell does is it passes it to somebody else. So in these oxidation reduction atoms or enzyme reactions, we are either storing it in the form of a B vitamin battery, or we're passing that electron on to something else, to the electron transport chain. So cell respiration utilizes oxygen, breathe in, and produces carbon dioxide. And just like photosynthesis, it's released in steps. Look at this equation. It's essentially the opposite of photosynthesis. So here we're not saying G3P, we're saying glucose, that molecule, is oxidized while the oxygen itself is reduced, and eventually at the end we end up with carbon dioxide, water, and some free energy. Cell respiration occurs in the mitochondria. So both plants and animals have mitochondria. We don't have chloroplasts, but if you're utilizing cell respiration, you're using that mitochondria. Just like photosynthesis, you have to be familiar with energy transformations and some of these terms. Let's see. In cell respiration, there are 4 main steps: glycolysis, which is actually breaking apart that sugar molecule; prep reaction, where we prepare that new substance to enter the mitochondria; the citric acid cycle, where we access those electrons; and the electron transport chain, where we dump those electrons off and essentially utilize them to make a ton of ATP. Now, some ATP is made throughout the way through substrate-level phosphorylation, but the main portion that makes most of the ATP is the final step in the electron transport chain. So glycolysis is where we break apart that sugar molecule, and we end up with 2 pyruvate molecules. And so we access those electrons, a couple ATP are made, but it takes ATP to get going. So we have to use ATP. Now this happens out in the cytoplasm. It doesn't utilize oxygen, because some bacteria can utilize it that don't have oxygen as their final electron receptor. And so it's a remnant of the cell ancestry without mitochondria. But essentially, it breaks apart sugar and gives us pyruvate molecules and a couple ATP. Now, pyruvate can't enter the mitochondria without being prepared first. It's like when you make food. Like, yeah, it's fine if you just make it bland, but it's better if you season it. You want to eat it more. So you could think the prep reaction is like preparing food to be eaten. We're taking pyruvate and we have to attach it to what's called CoEnzyme A. That's our seasoning. We're seasoning up this molecule. And so we create a molecule called acetyl CoEnzyme A or acetyl CoA, and that can go into the mitochondria. In the mitochondria, we have what's called the citric acid cycle, or it's also called the Krebs cycle, named after Hans Krebs. It's a system where we access the electrons, pull apart those electrons. But remember, when you access an electron, you can't just let it go. You have to store it. So when we pull electrons out of that carbon molecule, we have to store it in NADH and FADH. Those B vitamin batteries, just like we had NADP in photosynthesis, in cell respiration, we have NADH and FADH too in this case. Now, some carbon dioxide is released here, and we do make a couple ATP molecules, but it's mainly substrate level, which you could think of that as meaning not a lot. the ADP and Ps are there, so we might as well make some ATP, but it's not the main source. The main source is going to be here at the electron transport chain. So once we have enough ATP, or once we have enough NADH and FADH, they can bring those electrons to the electron transport chain, dump those electrons off. They're passed from protein to protein to protein, from cytochrome complex, and we're able to use that energy and these hydrogen ions from NADH and FADH2 and store them in the mitochondria and create a gradient. So you should probably remember this from chapter 7. We're going to be utilizing chemiosmosis. We can use those hydrogen ions to flow directly across ATP synthase and make a ton of ATP. This is called oxidative phosphorylation, and it makes a ton of ATP. So here are the steps: glycolysis out here, the prep reaction to prepare that pyruvate molecule, citric acid to access those electrons and form those B vitamin batteries in the electron transport chain to for us to pass along those electrons and then create a ton of ATP through chemiosmosis and oxidative phosphorylation. The rest of the PowerPoint goes through those steps piece by piece to allow you to get the details. And then we also discuss fermentation, what occurs inside cells if we don't have oxygen. So if you ever ran before, sprinted, sometimes you run out of oxygen and you start getting cramps. That's because your body has a primitive ability to keep running. Even though we might feel like we're out of energy, we can keep going. But this process is anaerobic. That means we're not consuming oxygen, we're out, but we have to make a little bit of ATP. It's not very efficient, but we can utilize lactic acid fermentation. And that buildup of lactic acid is what gives us those cramps. Again, I just go through step by step. And then we talk about these energy organelles, how the mitochondria is no longer just the powerhouse of the cell, like you learned in high school. It's now the site of ATP synthesis via oxidative phosphorylation, which is the step that makes a ton of ATP energy. Through cell respiration, that process, we don't only utilize glucose. There are other options. In this class, we just stick with that, but we can break down. Fats can break down, proteins can break down, and other substances can actually be put into that Calvin cycle. And not everything utilizes aerobic respiration. Some organisms are strict anaerobes. That means they don't use oxygen for their final electron receptor. If you ever seen let me think, like a canned good, like a can of food, and it's a little expired and it's bulging, you don't want to eat that because the Clostridium bacteria has built up inside of that and is releasing its toxic compounds, and you don't want that. And it can do so without oxygen in that sealed environment because it doesn't utilize oxygen as its final electron receptor. So keep that in mind for a post-apocalyptic scenario when we find a bulging, canned good, green beans or something. Don't eat it. Bulging means it's not good. And then I finally close off with endosymbiotic theory, talking about how those mitochondria, just like the chloroplasts, once upon a time were bacteria essentially that have been absorbed by these eukaryotic cells. Now they have this symbiosis, kind of like humans and our gut bacteria, and even the archaeans that live inside our gut bacteria. All of us are living in symbiosis. We can't live without each other. We require each other, but we're still our own thing. That's kind of what mitochondria are. Through time, they sort of lost all of their other functions, and all they are is just a machine, essentially, to pump out ATP. But they still have that remnant of their ancestry. They reproduce on their own, which is wild. We don't make them. We get them from our mothers. We might get a few from our father, but for the most part, we get our mitochondria from our mother's cells. And then our cells just make more. Or I'm sorry, mitochondria just reproduce. Our cells don't actually make mitochondria. They have circular DNA, bacterial ribosomes. It's wild. So look through that, and then my final slide again is ending on those stromatolites, which we talked about. So go through this. Let me know if you have any questions. I'm here to help.